Yeah, so what we're studying is sexual cannibalism in a native species of mantid. We know this occurs from time to time, but we're testing to see if the males can reproduce while they're being eaten or after they've been eaten. So we're raising mantids in our lab and breeding them as adults and testing and counting egg numbers and successful reproductive rates of the males that have been cannibalized versus the ones that have not been cannibalized. So it's a 2-year project because we have to raise the mantids up from babies with 3 different food sources. We have flies, crickets, and fruit flies. So the work is very intensive, and it takes a long time for the mantids to grow. This is our 3rd run through, so we're looking at a lot of data when we get done. And the relevance and importance in this situation is that this is a native species that is a top predator in the land and the area that it populates. So if we can determine one of the reproductive successes, we can calculate their survivorship rate in the wild, and that makes a big difference in, say, prairies or woodlands.
Okay, so what our research is on is, what we're studying is retinoic acid metabolism, and retinoic acid is an analog of vitamin A. And when you take in vitamin A, it gets cleaved into beta carotene, which starts off as retinal, which is involved in your vision processes. But retinoic acid is important in the body and cellular processes because retinoic acid serves as ligands for receptors, and when the retinoic acid binds to these certain receptors, it transcribes into like certain proteins that are important for cellular processes such as differentiation, apoptosis, cellular adhesion, and many other things. And so what we're looking at specifically is 9-cis retinoic acid and how it's metabolized.
You can see that both the TRONG and the keto with the 9-cis are inhibiting metabolites, which suggests that troglodytism can also be a cytochrome P450 inhibitor. And the reason why you would want this is because the genes that are transcribed, like the proteins that are made, help in diabetes to make your receptors for insulin stronger so that they work better so that you're not as diabetic. And so that's why you would want that inhibited, and that's why this is really important to us. But we plan to do more, different things with that with cell counts and running standards and stuff like that to make sure that what we see is what we actually think we're getting.